Welcome to the Deep Dive. Today we're jumping into something truly remarkable in medicine, Chimeric Antigen Receptor T-cell therapy, or CAR-T.
Yeah, it's been completely revolutionary, especially if you look at blood cancers. Things like B-cell lymphomas, ALL, multiple myeloma. CAR-T has, well, it's set a totally new standard.
A new standard is right. We're talking more than 10 approved products targeting CD19 or BCMA mostly, and they're delivering these incredible long-lasting remissions, sometimes even cures.
Absolutely. But, and this is the crux of our deep dive today, that amazing success story, it kind of masks a really significant ongoing challenge.
Which is?
Well, getting it to work nearly as well in solid tumors, you know, the big killers, breast cancer, lung, pancreatic. Progress there has been, let's say, limited, or as our sources put it, largely hindered.
Largely hindered. Okay, so that's our focus then. Why the struggle with solid tumors?
Exactly. Our mission today is to really unpack why. We'll look at the clinical progress that is happening, because there is some, and crucially dig into the, frankly, cutting-edge engineering strategies people are using.
Strategies to tweak the T-cells themselves.
Yeah, modifying their internal engines, figuring out how to deal with the tumor environment, and even changing how we actually make these therapies.
Okay, let's back up just a tiny bit. For anyone new to this, what is a CRR T-cell, fundamentally?
Right, so it's basically a type of white blood cell, a T-cell, that we've engineered in the lab. It takes the natural killing power of a T-cell.
Its ability to, seek and destroy.
Exactly. But it overcomes a really key limitation of our natural immune system.
And that limitation is?
It's called MHC restriction. Think of your natural T-cell receptor, the TCR, it needs a specific ID badge scanner.
Okay.
It can only see a cancer cell if that cell shows a little piece of itself, the antigen, on a special platform called the MHC. If the cancer cell hides that platform or doesn't use it right, the T-cell is blind.
So the CRR, this chimeric antigen receptor, it's like giving the T-cell night vision goggles. It doesn't need the ID badge scanner anymore.
Precisely. The Bayer-TLR is this synthetic receptor we add to the T-cell. It has an outside part, often derived from an antibody called an scFv, that can grab onto the cancer antigen directly. No MHC needed.
Huh?
And it's modular. You've got that grabbing part, a hinge, a bit that sticks through the cell membrane, and then crucially, the inside parts. These provide the go signals, usually the T-cell's own signaling chain, TCR-zeta, plus what we call a costimulatory domain, like CD28 or 41BB.
So it gets the full go, attack signal when it binds.
That's the idea. Full activation.
Makes sense. And that worked brilliantly for blood cancers, where you have targets like CD19 everywhere. But take that engineered killer, put it up against a solid tumor, and bam, brick wall.
That's a good way to put it. Which brings us neatly into section 1, the core roadblocks in solid tumors.
What's hurdle number 1? Our sources call it the triple threat.
Threat number 1 is a big one: on target, off tumor toxicity. OTOT for short.
Yeah.
Well, in blood cancers, targeting CD19 wipes out healthy B cells too, but we can manage that. With solid tumors, the targets are usually tumor-associated antigens, TAAs.
Associated, not specific, so they're also on healthy cells.
Exactly. Maybe at lower levels, but they're there. If your CAR-T is too aggressive or the target is on vital organs, you can get devastating toxicity. You kill the tumor, but you severely damage the patient's healthy tissues. It's a huge safety risk.
A major clinical tightrope to walk. Okay, threat number 2, antigenic heterogeneity.
Right. Think of a solid tumor, less like a uniform blob and more like a bustling, chaotic city. The cancer cells inside aren't all identical.
Okay, to vary.
Hugely. And they mutate, they change. Some cells might have lots of your target antigen, others very little. Some might lose it altogether to hide from the immune system. This is antigen escape.
So finding a target that's reliably on all the cancer cells is tough.
Incredibly tough. You need something stable, highly expressed, ideally on, say, 80–90% of the tumor cells. Otherwise, you just kill off some of them and the resistant ones grow back.
And the third threat, the one you mentioned, is maybe the biggest deal: the immunosuppressive tumor microenvironment, the TME.
Absolutely. This is where solid tumors are fundamentally different from blood cancers. The TME isn't just some free-floating cells, it's a physical place.
Like a fortress.
Kind of. You've got dense connective tissue, this extracellular matrix that physically blocks T cells from getting in. You've got messed up blood vessels that don't let immune cells traffic properly.
So just reaching the tumor cells is hard.
Very hard. And even if they get there, the chemical environment is incredibly hostile. It's packed with suppressive immune cells like TREGs and MDSCs and flooded with molecules that actively shut T cells down. Things like TGF-beta, adenosine.
Okay, so let me get this straight. The CRA T cells have to first navigate this physical maze, push through barriers.
Right.
Then, once inside, they're hit with this constant barrage of stop signals, this chemical warfare.
Precisely. And what happens when a T cell faces that kind of chronic stimulation combined with constant suppression?
It gets worn out.
It gets exhausted. T cell exhaustion. It's a natural biological program, actually, meant to prevent overactive immune responses. But in the TME, it means our CRT cells just fizzle out.
They burn out before they can finish the job.
Exactly. Their function declines, they stop proliferating, they don't persist long enough to clear the tumor. That's been a massive barrier to durable responses.
Wow. Okay, so OTOT risk, antigen heterogeneity,
Yeah.
and this incredibly hostile TME, that's a formidable set of challenges.
It really is.
But,
Well,
and there's always a but in science.
Absolutely.
We are seeing some encouraging signs, right? Let's look at some specific clinical snapshots, phase 1 trial data,
Yeah.
focusing on particular TAAs.
Yes, absolutely. Despite the hurdles, there's definite progress. Let's take hepatocellular carcinoma, HCC, liver cancer. A really promising target there is glypican-3 or GPC3.
Why GPC3?
It's overexpressed in like 70 to 80% of HCC cases, but pretty low on most healthy tissues. That helps with the OTOT issue. And there's one therapy, CCR031, which is an armored CAR-T.
Armored, meaning it has extra engineering to resist the TME.
Exactly. In this case, designed to resist TGF-beta. In a phase 1 trial, this therapy achieved a disease control rate DCR of over 90%.
90%. That means stopping the tumor from growing or shrinking it in 9 out of 10 patients.
For a period, yes. And the objective response rate, meaning significant tumor shrinkage, was 50%. In advanced, hard-to-treat liver cancer, those are really striking numbers.
That is striking. What about gastric cancer?
There, a key target is Claudin 18.2 or CLDN 18.2. It's a protein normally tucked away in stomach lining junctions, but it becomes exposed on the surface of many gastric and pancreatic cancer cells.
Interesting. And the results.
Pooled data from trials using a CAR-RT called CT041 targeting CLDN 18.2 showed a DCR, again, really high, almost 92% in advanced gastrointestinal tumors. The ORR was around 39%. Again, showing these CARs can work if they engage the target.
Okay, impressive DCRs. Maybe the most dramatic field is brain tumors, gliomas.
Yeah, gliomas present unique challenges, like getting the RT cells past the blood-brain barrier often requires direct locoregional delivery, injecting them right into the brain or spinal fluid.
High risk, high reward.
Potentially. Targeting B7H3 in recurrent glioblastoma, a really aggressive brain tumor, showed incredible results in one phase 1 trial, an 83% overall survival rate. Median OS was over 20 months.
That's huge for GBM.
It is. And another target, GD2, used in CAR-T therapy for children with a devastating type of glioma called H3K27 mutated diffuse midline glioma, led to complete durable responses in some patients. Truly game-changing potential there.
Okay, let's do a quick recap here. We've got solid tumor targets like GPC3 in liver cancer, CLDN 18.2 in gastric, B7H3 and GD2 in brain tumors. They carry that OTOT risk. The TME fights back hard.
Exactly.
But these early clinical results, especially the high DCRs and the survival data in glioma, show that, wow, this can work. Efficacy is possible.
The proof of principle is there. We can kill solid tumors with the CAR T cells. But that T cell burnout, the exhaustion, the persistence issue, that's the real bottleneck preventing widespread success.
Which perfectly sets up our next section. If the T cell is burning out, how do we fundamentally re-engineer it? How do we make it tougher, more resilient?
Exactly. This brings us to section 3, engineering strategies. And let's start inside the T cell itself, enhancing its persistence and fighting exhaustion by tweaking its programming, specifically the transcription factors, or TFs.
TFs control which genes are on or off, right? So you're changing the cell's core instructions.
Precisely. We want to push the cell towards a long-lived memory state, not a short-lived effector state that burns out quickly. A key TF for memory is FOXO1.
Okay.
Overexpressing FOXO1, making more of it, helps program that durable memory phenotype. It improves persistence and efficacy. Think of it like installing better endurance software.
Got it. So you boost the staying power TFs. What about the burnout TFs?
Yes. We also need to hit the brakes on exhaustion. Certain TF families, like NR4A and TOX, get cranked up when the T cell's chronically stimulated in that hostile TME. They actively drive the exhaustion program.
You want to remove them.
Exactly. Using gene editing, like CRISPR, scientists can knock out these specific TFs. And the results in preclinical models are clear. Knocking out NR4A, for instance, reverses T cell dysfunction and leads to better tumor control. It's like removing the software that tells the cell to give up.
Molecular reprogramming. Cool. Beyond the programming, what about fuel? You mentioned the TME is chemically toxic. How do you make the CAR T cell more metabolically robust?
That's metabolic overhaul. The TME is often low in nutrients T cells need, like glucose, and high in suppressive metabolites, like adenosine. We need to make the CAR T fitter. Several ways. One example is targeting proline metabolism by overexpressing an enzyme called PRODH2. This seems to enhance killing and support memory. Another big one is adenosine resistance.
You mentioned adenosine earlier as a suppressive signal.
Right. It signals through the A2A receptor on T cells, so simple strategy. Genetically delete the A2A receptor, knock it out.
The T cell becomes deaf to adenosine.
Exactly. It resists that suppression, keeps producing key anti-tumor molecules like interferon gamma and TNF, and maintains function without sacrificing persistence. It's a really elegant metabolic edit.
Okay, better programming, better fuel efficiency. Now let's tackle the battlefield itself, the TME. First up, just getting there. Improve trafficking and homing, giving the CAR T better GPS.
Yeah, the tumor's messed up vasculature and dense structure make it hard for T cells to physically get in. One strategy is to engineer the CAR T cells to express chemokine receptors, like CXCR2.
Chemokines are like come hither signals secreted by tumors.
Some are, yes. So expressing the right receptor helps the CAR T cell follow those signals, like a heat-seeking missile, guiding it into the tumor mass more effectively.
And you mentioned a really dramatic example earlier involving chemokines.
Ah, yes, the IL-7 and CCL19 combination. These aren't receptors, but molecules the CAR T cell itself is engineered to secrete. They basically create an inflammatory beacon, attracting not just more CAR T cells, but also other helpful immune cells into the tumor.
Creating a better immune hub right inside the enemy territory.
Exactly. And that was linked to that one stunning case in HCC where the tumor just vanished in 30 days. Right. Shows the power of overcoming the trafficking barrier.
Okay, better GPS. What about breaking down the physical walls, that dense matrix, the ECM?
For tumors rich in that matrix, like some pancreatic or breast cancers, you can arm the CRT cells with matrix-degrading enzymes. The classic example is heparinase.
So the CRT cell essentially becomes a little bulldozer.
Yeah, that's a good analogy. It expresses heparinase, which chews through components of the ECM, clearing a path for itself and potentially other immune cells to infiltrate deeper and attack the tumor cells.
Clever. And finally, resisting the chemical attacks within the TME.
Right. Fighting those suppressive signals. We talked about adenosine. Another huge one is TGF-beta. It's a potent immunosuppressant found in many solid tumors.
So, similar strategy. Make the T cell deaf to it.
Pretty much. You knock out the TGF-beta receptor 2 on the CAR T cell. It can no longer receive that stop signal, which helps prevent exhaustion, especially in challenging TMEs like liver and pancreatic cancer models. This is often combined with other edits like knocking out PD-1, another key immune checkpoint.
Wow. Okay, that's a whole suite of sophisticated engineering, internal tuning, metabolic boosts, GPS upgrades, bulldozers, and signal jamming.
It's becoming a multi-pronged approach. You need to address several of these issues at once.
Which leads us to Section 4, thinking about the CAR receptor itself and how we make these things. We need smarter receptors, right? To avoid problems like that early burnout.
Definitely. A key issue with some earlier CR designs was tonic signaling.
Like that.
It's basically the CAR receptor being slightly on all the time, even when it's not bound to the tumor antigen. It's like a car engine idling too high.
And that constant low-level signaling contributes to exhaustion.
Yes, it pre-exhausts the T cell. So current research uses sophisticated tools, even AI-driven platforms like CAR Toner, to analyze the receptor structure and fine-tune it, reducing those charged areas that cause unwanted signaling.
Tweaking the design.
And also incorporating different signaling components. For instance, adding specific motifs, like the EB6I motif from the CD3 epsilon chain, seems to improve the quality of the signal, making the CAR T more potent, especially against tumors that don't have a lot of the target antigen.
Interesting. And then there's a more radical redesign, STAR T cells. What are those?
STAR stands for a synthetic T-cell receptor and antigen receptor. It's a really clever hybrid. It uses an antibody-like part to bind the antigen, like a CAR.
Okay.
But it's structurally designed to plug into the T-cell's natural TCR signaling machinery.
Instead of having its own separate signaling domains.
Exactly. The result is signaling that's slower, more moderate, more like a natural T-cell response. And this seems to give STAR T-cells superior sensitivity, especially for tumors with very low levels of antigen. It's a potential leap forward in function.
Fascinating. Okay, better function is great, but safety is paramount, especially with that OT risk. We need clever safety switches or logic gating.
Absolutely critical. One approach is affinity tuning. You deliberately engineer the CAR's binding domain to have lower affinity to not grip the target quite so tightly,
Why would you want it to bind less well?
so it can discriminate. The idea is it will only trigger a strong killing signal when it encounters high levels of the antigen as found on cancer cells, but ignore the low levels found on healthy tissues. It dials in the sensitivity.
Makes sense. And for even more precision.
You use A&D gated logic. This is like needing two keys to open a lock. The CRA lens cell is engineered to only fully activate if it simultaneously recognizes two different antigens on the cancer cell surface.
So if it sees antigen A or antigen B, nothing much happens.
Right. Or maybe a weak signal. But if it sees antigen A, AND, antigen B together on the same cell,
Full activation, killing sequence initiated.
precisely. This dramatically increases specificity and reduces the risk of attacking healthy cells that might only express one of the targets. It's a powerful way to combat OTOT. Plus, you still have backup options like inducible suicide switches if severe toxicity occurs unexpectedly.
Amazing molecular control. Okay, final piece of the puzzle, and maybe the most impactful for patients worldwide, manufacturing innovation,
Yeah.
making these therapies accessible.
This is huge. Traditional CAR-T production is complex, expensive, and patient-specific. It takes weeks. We need faster, cheaper, more standardized methods.
How do we get there?
One big shift is moving away from using viruses to insert the CAR gene, which integrates somewhat randomly and carries some risks. Now, the field is rapidly adopting non-viral gene editing, especially CRISPR-Cas9.
For more precise insertion.
Exactly. You can use CRISPR to cut the T cell's DNA at a specific predefined safe spot, like the TRAC locus, which disrupts the native TCR, or the PDCD1 locus, which knocks out PD1 at the same time, and then slip the CAR gene right into that precise location. It's cleaner, potentially safer, and leads to a more uniform product.
Standardization. And the ultimate goal for that.
The off-the-shelf revolution. Allogeneic CAR T cells.
Meaning cells not from the patient, but from healthy donors.
Yes. You take T cells from carefully screened healthy donors, engineer them in large batches, potentially making hundreds of doses from one manufacturing run.
What are the advantages?
Massive potential advantages: standardization, much lower cost per dose, scalability, and crucially, immediate availability. Patients wouldn't have to wait weeks for their own cells to be engineered. Plus, donor cells might be functionally healthier than cells from heavily treated cancer patients.
Is this actually working in solid tumors yet?
The early signs are incredibly promising. There was a major milestone recently. The first reported complete response in a solid tumor patient, someone with renal cell carcinoma, treated with an allogeneic CD70 targeted CAR-T called CTX130. It shows it's feasible.
Wow. Okay, so wrapping this all up, the journey for KA in solid tumors has been tough, facing these huge biological hurdles. But what we're seeing now is this incredible convergence. Sophisticated T-cell engineering to boost persistence and metabolism, clever strategies to overcome the TME's defenses, smarter receptor designs like STAR-T, safety gates.
And now, manufacturing breakthroughs like precise gene editing and the real possibility of off-the-shelf allogeneic therapies. It's tackling the problem from every angle.
It really feels like we're turning a corner, moving from identifying the problems to engineering concrete solutions.
I agree. The pace of innovation is stunning. We're proving we can manipulate these cells to be smarter, tougher, safer, and potentially much more accessible. The future feels significantly brighter than it did even just a few years ago.
So we'll leave you, our listeners, with a final thought to chew on. If these off-the-shelf allogeneic CAR-T therapies are proving feasible, even showing efficacy in tough solid tumors like kidney cancer, and they promise dramatic cost reductions and faster access, what's the next big hurdle? If the science and manufacturing are aligning, how do we ensure this powerful technology actually reshapes the global standard of care and reaches every patient who could benefit, no matter where they live or what resources they have? That access question feels like the next frontier.